Good morning and welcome to our frameworks on emergency use authorizations. Today we have a great group of doctors and that are going to give us some more information about vaccines, therapies, and diagnostics under emergency use authorization. So welcome everyone. I'd like to welcome our president and CEO of BioCrossroads, Patty Martin, to kick off the show. Welcome, Patty.
Thanks so much, JoBeth. Welcome, everyone. We are so delighted to have a very diverse audience with us today for this topic. This is actually the third session we've had on the topic of COVID or vaccines. And if you haven't had a chance to participate or listen in to what was shared during those sessions, they're available on our website. So we would be very happy for you to go back and take a look at that content. Today, we're very excited because we're going to hear from a number of experts on the topic of emergency use authorizations and the difference between mRNA and conventional vaccines. These are words that are being thrown out there a lot. And even with 26 years at Lilly, I can tell you I had to do my research to understand what the terms meant. So we think that an informed audience is one that's educated and can share the word on the facts around these two topics. So we're very thrilled to have you all join us today. And at the end of this frameworks, we want you to enjoy having the opportunity to ask questions. And so before I introduce the speakers, I'd like to inform you that we're going to use the chat function today on Zoom. So please, please, please enter your questions into chat because we have some fabulous experts that will be able to help inform you, and no question is off limits. So again, our intent is to inform you, our audience, and hopefully you then can share the word with others. This framework is being recorded and we will have it available on our website shortly after today's session. So with that, I'm going to step back and introduce our first speaker. Dr. John Christensen is from the IU School of Medicine. He is a pediatric infectious disease specialist who will talk about the importance of vaccines and give us a practical understanding and definition of emergency use authorizations. Welcome, Dr. Christensen.
Good morning. Thank you for the invitation. I guess we'll go with our first slide, right? So that's my affiliation there. Next slide. I have no financial relationships with any of the manufacturer of any of the products or vaccines that we're going to talk about today. So next slide. Now, I've been a pediatric infectious disease doctor for over 30 years. So that tells me that tells everyone that I'm on the older side, and I'm the elder physician in our section here at Riley. So I've had the opportunity to see seeing a lot of the vaccine preventable diseases that so much we vaccinate against now these days, and people are wondering, you know, why we vaccinate since these diseases are gone. But I remember days back in the 1980s where we had Haemophilus influenza infections, patients with those type of infections in the hospital all the time with all kinds of different problems, including meningitis and including some of these children dying of these infections. The last two cases I saw of Haemophilus influenza type B meningitis were in 2001 in 2 unvaccinated children. Soon after I arrived at the University of Utah back in 1987, there was a huge outbreak of measles in the state where there were about 600 cases. Where the first 2 initial cases were undiagnosed. But now these days, most physicians that are trained in our hospital have never seen a case of measles and perhaps will never see one. The same thing, varicella, mostly gone thanks to vaccination, but we used to have people who would die of streptococcal sepsis and cellulitis due to complications with varicella. I still remember a child who developed a chronic devastating and fatal condition seen with measles, SSPE, subacute sclerosing panencephalitis, a condition that is, I don't think physicians in the United States have seen a case in many places for years, but that was soon after I arrived here in Indiana. So meningococcus is another good example, and then we have cases of pertussis. So a lot of these conditions have been made not non-existent, but significantly reduced thanks to vaccination. Next slide. Now you look at measles, you know, in the last few years, still we see measles in the United States because of importation into the United States with travelers coming from other places. Next slide. Varicella, this is the kind of problems that we used to encounter, you know, disseminated disease and immunocompromised hosts. Next slide. Now, rotavirus, a condition that we saw our share of diarrheal disease here in the United States, resulting in hospitalizations and very rarely mortality. However, when you look at the effect of vaccinations over diarrheal diseases caused by rotavirus, let's say in Mexico, the introduction of rotavirus vaccine resulted in a significant reduction in mortality. And this has been the case with vaccinations throughout the world. Next slide. This is Haemophilus influenza type B. Thanks to vaccination, you can see there that nationwide, the number of cases are significantly reduced and very rarely seen. Next slide. The same thing with invasive pneumococcal, thanks to the vaccine that we give to young infants, the incidence of meningitis and bloodstream infections that is seen with this pathogen is not only significantly reduced in young children, but also had an effect over reducing infection in older individuals, thanks to reduction of colonization with this particular bacteria, and vaccination also of adults with this type of vaccine. Next slide. Now, when you look at the graphs, this comes from the CDC, and you know, I'll let you look at it in greater detail. But you can see there that for all the conditions for which we have vaccinations, we have seen a reduction of disease thanks to vaccination. And you can see there some good examples. In some cases, like in varicella, there was a 98% reduction. In other conditions, it may be a little bit more modest, a reduction. Next slide. But for example, when you look at conditions like smallpox, we have no smallpox around the world thanks to vaccination. When you look at the cases of diphtheria reported in the United States, you see there that there's a big zero there because that is non-existent in the United States. Pertussis, we still have a challenge with that. Polio has been eradicated from the Americas, and there's only 2 countries in the world that have some transmission of polio right now, Pakistan and Afghanistan. And you can see there the significant reduction seen with measles, mumps, rubella, and Haemophilus influenza. Next slide. And we have to thank vaccines for this. Now, I like to put this is everything perfect, right? The challenge that we have as clinicians and for all of us who work in healthcare is that there is this misconception that through vaccinations, all these conditions, all these infections are now gone. So the common hesitation for vaccination, one of the most common reasons that why people don't want to get vaccinated or don't want to have their children vaccinated is because they think that these conditions are gone. So why should we vaccinate against them? As a result of that, in communities that have hesitated vaccinating, we've had outbreaks of measles. We've had some outbreaks of mumps as well. And we have exposures that occur in hospitals with varicella, and we have other conditions that still need to be controlled through vaccines, right? We know that all vaccines are not perfect, right? We know that in spite of having good penetration of the use of measles, mumps, rubella vaccine, the so-called MMR, we still have outbreaks of measles, of mumps, because we know that the vaccine still underperforms in that type of protection. But for example, conditions like human papillomavirus and influenza, we still have challenges in convincing people to get those vaccines. And not only the hesitation over the fact that these conditions are probably rare, but at the same time, people are concerned about side effects. Next slide. So these kind of challenges, we're now seeing this even with the penetration and getting people to vaccinate against coronavirus. And so we have to convince people that we're dealing with fatal diseases. We also have to factor in that not everybody is symptomatic, but those that are symptomatic can get really ill with this. And then we have challenges in high-risk communities in African American communities and Hispanic communities, but also we have high risk groups like the elderly and children or the immunocompromised that we would like to get vaccines to try to prevent disease. So obviously we're all looking for vaccines that are safe with no side effects, and industry has done pretty good in providing us vaccines that meet those criteria. However, we still have challenges because we have to be very good in convincing people that this is the best approach in preventing disease and keeping people healthy. Next slide. Now, we will talk a little bit about EUAs. People will go into greater details about EUAs, emergency use authorizations for not only diagnostic tests, but for vaccines. You can see that this term has been used quite a lot, right? And vaccines have been approved, drugs have been approved using EUAs, diagnostic tests have been approved. Next. Now the EUAs, you can see this is one of the kits that has been approved for EUA by EUA. We won't go into great detail about that, but next slide. But the most important thing is that people understand where this EUA, most of us probably, unless we've been involved in pandemics or major outbreaks or major public health emergency in the United States, have perhaps never heard about an EUA. And the EUA has been something, it's a part of our FDA and the Federal Food and Drug Cosmetic Act that allows, in the case of a public health emergency, for somebody to declare this emergency. You can develop vaccines, you can develop diagnostic tests, you can develop treatments that fall into this criteria. And because of the emergency, they can be used in humans. Obviously, you have to qualify certain things. You have to show some safety. You have to show clinical utility. But the important thing is that this act allows healthcare authorities to make this designation and allow the use of these products, vaccines, and tests and treatments in patients. This emergency is not declared by anybody. It's declared obviously through the government, the president, and through the commissioner of the FDA. Next slide. Now, the most important thing to keep in mind is that these products that are authorized under use of the EUA are not approved drugs by the FDA. They don't go through the same approval process. However, they are, safeguards where evaluate these products and they evaluate these vaccines and they evaluate these treatments. So it's not that somebody just declares that we're going to have an EUA today. No, they has to go through a process. And you can see there that people get together and they decide that an emergency is important. And then you have the Secretary of HSS declare this emergency through the FDA commissioner. And there is consultation with all kinds of different groups and experts to determine if these can happen or not. And when they talk about vaccines, you'll see the EUA process for there. Next slide. The important thing is that you can see there are all kinds of different things that have been approved under EUAs, treatments, therapeutics, diagnostic tests, vaccines, and the mRNA vaccines are a good example of that. Next slide. And the important thing is that all kinds of other products, products that are used to sterilize masks and PPE, even ventilators could go through this process as well, depending on what the emergency is. Next slide. Now, the important thing to keep in mind is that because there's been a lot of debate is what does the EUA allow? Well, it allows to use these products. The debate comes about, can you force people to get the vaccine? Can you force people to get the treatment? And that's where there's a lot of discussions among legal individuals talking about this. But for example, you can see there that there are going to be certain things that have to be met. But generally, many people would say that under EUAs, it's very difficult for industry or hospitals or companies to enforce the mandatory use of certain medications or specifically vaccines, like we do in healthcare with influenza vaccine, which is an FDA approved vaccine. Next slide. The important thing to keep in mind is that these EUAs are not permanent, right? Once the FDA commissioner and the Secretary of Health and Human Services and other authorities determine that the emergency is over, well, companies are required to reconsider the EUA and to seek official approval through the FDA. And that will be the case with vaccines because they're likely to stick around for a long period of time. But when it comes to other treatments and other medical devices, if the company would like to keep those in place, they would have to seek official approval. And that is just the normal process. So right now, we're in the middle of a pandemic, so I would not be surprised that you will see more products coming out under the EUA, and with time, we'll see how much we'll keep them, okay? So I'll end there, and the other speakers will have plenty to talk about vaccines and therapeutics, as well as diagnostic testing. Thank you very much.
Thank you, Dr. Christensen. Our next speaker is Dr. Laura Parnas from Roche Diagnostics. Laura is the Senior Scientific Affairs for Roche Diagnostics. Please welcome Laura.
Good morning, everybody. It's a pleasure for me to be here today to present to you the perspective about the emergency use authorization for diagnostic testing. So let's go to the next slide, please. My disclaimers: I am an employee of Roche Diagnostics in the non-commercial division of Medical and Scientific Affairs. And the information that I will present today is current as of January 20th of 2021. Next slide, please. As Dr. Christensen said, there are several FDA regulatory pathways for new medical devices. So you may have heard different terminology depending on what type of device is getting through the FDA regulatory pathway. So here is just a summary of what we encounter in the diagnostic space. We can see different classes according to the risk, and then that there are different pathways for the submission to FDA. So we have exempt devices, and those ones are called FDA registered or listed, and they usually take about 3 weeks as a process. And then we have Class 2 devices, which have moderate risk, and usually these ones encompass a 510K submission. And usually we call this products FDA cleared. And a typical submission takes an average of about 5 months. And then we go into Class 3 devices, high risk or no predicate, and these ones are the pre-market approval, PMA, and those ones are the devices that are FDA approved. And this process is a little bit longer. It's a very thorough submission, so it takes an average of 18 months. And then we have these alternative pathways, and Dr. Christensen very clearly and eloquently described what the EUA looks like. And these alternative pathways are in very special circumstances for humanitarian purposes, product development, etc. And then the EUA, the emergency use authorization, falls under this alternative pathway for FDA. Next slide, please. Again, just a little bit of a reiteration here. What is the emergency use authorization? Allows the FDA to help strengthen the nation's public health protections. And this is again given from the Federal Food, Drug, and Cosmetic Act, where the FDA commissioner, under the declaration of an emergency, allows unapproved medical products or unapproved uses of approved medical devices to be used in this emergency. And then for diagnostic testing, we follow the policy for coronavirus disease 2019 for all of our COVID diagnostics. And the FDA has actually updated this policy a couple of times since the beginning of the pandemic. And today we are following the policy that was issued on May 11th of 2020. Next slide, please. A little bit more about the EUA. Again, this is meant to specifically address this medical need. And then in terms of what the FDA expects from the manufacturer is that we will continue the path to either a 510K clearance or a PMA approval so that the product can be used after the emergency period ends. And then in this case, for COVID-19, the goal or the objective of the emergency use authorization is to prevent, diagnose, or treat COVID-19. So the devices that are granted EUA by FDA will be either to prevent, diagnose, or treat COVID-19. And they have to meet 4 criteria. It's a serious or life-threatening disease or condition. It's evidence of effectiveness, risk-benefit analysis, and there are no other alternatives. Next slide, please. And I'm going to ask you to build it one more time so we can see the entire slide. So in terms of the SARS-CoV-2 diagnostic test overview, we have two situations where we want to use different types of tests. The question comes, is somebody being infected with the virus? So we want to have a diagnostic test that detects the virus. And you have heard this on the news. It's been really highlighted everywhere in terms of the diagnostic testing. The PCR or the molecular test, which can be done either in the central laboratory or we also have antigen tests that actually, instead of detecting the genetic material from the virus, they actually detect the proteins from the virus, and they also can be done either at the central laboratory or at the point of care. And then we also have tests that will prove if somebody has recovered or if somebody has been exposed to the virus. And in this case, with the vaccines that are available, they are able to measure the vaccine response. And it's because the vaccines trigger the development of antibodies. So either you will have antibodies because you have been exposed naturally to the virus, you have had an infection with SARS-CoV-2, or you have received the vaccine and we're able to detect the immune response. And again, this antibody testing can be done either at the central laboratory or at the point of care. Next slide, please. But not all COVID tests are created equal. So FDA has been really overwhelmed with the amount of submissions for emergency use authorization in all different types of areas, right? For diagnostics, for treatment, and for PPE as well. So there are now hundreds of emergency use authorized diagnostic tests, both for diagnosis and also for detection of antibodies in different kinds of format. And then FDA also has evolved their knowledge about how to deal with these submissions. And they've also become a little bit more strict in terms of what is the type of information and data that needs to be submitted, and in order for them to actually provide a review and grant the authorization for this test to be in the market. So initially there was an umbrella process by which many different companies were able to distribute their assays, their tests, but FDA a couple of months later decided to revoke this umbrella and have everybody who had the intention of distributing tests to actually perform an emergency use authorization submission. But anyway, not all the COVID tests are created equal. There are certain things that we as manufacturers have to take into account. The goal is to have no cross-reactivity with any other coronaviruses or other viruses, right? So we need to have a test that is very specific for SARS-CoV-2. Also that it has optimal clinical sensitivity and specificity according to the test setting. So you don't want to have a false result any time, whether you're getting diagnosed or whether you're getting measured your antibodies. And then reproducible results, which means that the results that you're getting in different times or occasions are exactly the same and they don't change over time. So we want to have a test that is a high quality, that it also targets adequate proteins. You've been hearing a lot about these mutations that are happening, and also that they have optimal clinical performance, that they will do what the test is meant to do. Diagnose it when it has to diagnose, and actually test for the presence or absence of antibodies when that is required in each one of these diagnostic clinical settings. Next slide. And I'll just tell you a little bit about what our journey looks like for a normal test, diagnostic test that we want to bring to the market. It usually takes us, I would say, an average of 6 to 8 years. We look at whether there is an unmet need, and then we have a disease area strategy. We do the research, we develop the assay, and then we finally have to do a clinical trial. FDA requires that we have to do a clinical trial to demonstrate that the test performs for the particular intended use, or the use that the test is meant to do. And this takes an average of 6 to 8 years depending on the test. And next bill, please. And in the case of COVID, we have been able to actually compact this process into 6 to 8 weeks. It's been, at Roche Diagnostics is unprecedented. We have been able to develop all these assays in record time, given the gravity and the emergency of the pandemic. And then again, the EUA process allows us to be able to provide the data that we have generated in that period of time to demonstrate that our assays are safe and effective, they're high quality, and then they provide the right clinical information. Next slide, please. So I'll show you just a timeline. You'll see several timelines. This starts in January of 2020 and then ends in Q1 of 2021. January 30th, the declaration of the international global emergency by the WHO. And on February 1st, we started our work in the wet lab in Pleasanton, California, where our PCR tests are developed. And then on March 11, we actually submitted our SARS-CoV-2 assay for emergency use authorization, and it was granted on March 12th. It was the first PCR molecular assay for SARS-CoV-2 in the US. And on March 13, we started shipping the tests. And then in May, we received EUA for the first antibody assay against SARS-CoV-2. This assay was developed in 42 days. And then in June, we were granted EUA for interleukin-6 as a test to manage COVID-19. And then in September, we received EUA for our point of care as a test, and also for the combination of COVID and flu. And then last November, we received our last EUA for another antibody assay that we have. And then we are also working on a couple of point of care assays and an antigen central lab assay. So as you can see, it's going to be a total of 9 different tests in a little bit of about a year that we have been able to develop. Next slide, please. And then I just wanted to leave you with some thoughts. We have experienced, this is very unique in terms of this pandemic. We have seen the speed of the science develop in front of our eyes. It's really very hard to keep up with the literature. Things are coming really fast and furious at us. We have answered many questions, but there are still many other questions that are pending that we continue to learn every day. So I try to remind everybody that this is brand new. It's been a year. We have learned a lot about it, but there's still a lot more to learn. Stay tuned. And then other piece of advice that I provide is check your sources. There are so many great scientific resources out there that are all free to access. Main journals have opened up their subscriptions to make all the COVID-19 resources free. So a lot of resources out there for you. And then here I leave you with some of the questions that we've been asked, and then we know they're out there. Questions about protection and immunity. How long is our immunity going to last? How long are the vaccines going to be effective? And then how does this disease severity correlate with the immune response, and what happens in asymptomatics? So still a lot to learn, but reminding everybody that we have learned a huge deal and we continue to learn every single day. So thank you very much. I'm gonna end there.
Thank you, Dr. Parnas, for all that great information. Our next guest is Dr. Kathryn Ramseyer. She is Senior Director of Global Regulatory Affairs at Eli Lilly Company. Please welcome Dr. Ramseyer.
Thank you, and thank you for the opportunity to be here today to talk about emergency use authorizations for treatments for COVID-19. Dr. Christensen highlighted the general requirements to invoke an EUA, and Dr. Parnas covered very nicely the criteria for diagnostics. I'll be talking about those requirements for treatments, and we'll provide some insights into the differences between emergency use authorization and full approvals. And we'll try to show you a little bit more illustrative pictures about how that looks in the clinical setting using some of our Lilly experience as examples. Next slide, please. We covered this at a high level earlier, but I did just want to spend a minute on when FDA can authorize a treatment for emergency use. And this is guided by the guidance that's shown here on the left of the slide. There are 4 triggers that can lead FDA to allow an emergency use authorization. The first is that a domestic emergency can be determined by the Secretary of Homeland Security. The second, a material threat as determined by the Secretary. A military emergency as determined by the Secretary of Defense. And finally, a public health emergency as determined by the Secretary of Health and Human Services. And that's what happened on February 4th of 2020 when the Secretary announced a public emergency around COVID-19. Next slide, please. One overarching principle when pursuing emergency use authorization is that we don't actually stop with an EUA. It is an expectation of FDA that we will pursue a full approval for these products. Given that, the development program actually looks quite similar. The big difference is it is on an expedited and abbreviated path. So let's start with the evidence standard. For an emergency use authorization, a product may be effective. And this is an appropriate standard. The standard is judged as the known and potential benefits may outweigh the known and potential risks. This is appropriate because there are no alternatives that are adequate, approved, and available to treat the condition under consideration. For a full approval, we have to demonstrate effectiveness using a standard called substantial evidence. And that really is when we have to generate sufficient data that there's a positive benefit risk ratio, meaning we have to have sufficient data to prove the benefits of the medicine outweigh the risks. And this is typically done using at least 2 adequate and well-controlled phase 3 clinical trials to confirm findings from earlier stages of development. Next, let's talk about FDA engagement. And in my experience, this is where the biggest change has been seen and where one of the biggest impacts is. Under an EUA, we are able to obtain advice and review from FDA in a matter of days. They have actually reviewed, as you saw on earlier slides, they've reviewed EUA applications in days and often in weeks. For an approval, this is a much longer process. Just to gain advice or a meeting with FDA, we have to wait at least 30 days, usually more like 75. And when we talk about seeking full approval for a product, that takes at least 8 months, normally 12, depending on the disease state and the medicine that we're talking about. Next, let's talk about the data package. So as shown here, there aren't significant differences in the data package. For an EUA, we have to demonstrate the mechanism of action of the product. We have to provide all of the non-clinical and clinical safety and efficacy data. And finally, the CMC package, which is the data package that supports the manufacturing of the product. For a full approval, we provide the same data. The big difference here is that it's much more extensive. One contrast is the CMC package. For an emergency use authorization, we must demonstrate that we can consistently produce a safe and efficacious product. But for a full approval, we must go through a full process validation, which is a presentation in and of itself. Likewise, from a clinical perspective, often we can see emergency use authorization granted based on a single phase 2 trial that meets that evidence standard of may be effective. Whereas for a full approval, we do have to show substantial evidence. And again, that is typically through at least 2 adequate and well-controlled phase 3 clinical trials. Next slide, please. So next, let's talk about labeling. And this is the information that accompanies the product to inform prescribers and patients about the information available. For an EUA, we use a document called a fact sheet. And this looks quite a bit like the full prescribing information that I'll talk about in a moment. The difference is that not only does this tell what we know about the product, it tells what we don't know and the limitations of the available data to ensure full transparency for prescribers and patients. There are 2 fact sheets generated for a product under an EUA. One is for the healthcare professional, and the other is for the patient, parent, and caregiver. And that fact sheet must be provided to the patient prior to administration of the product. For a full approval, we produce what's called the US prescribing information, which is often accompanied by a patient package insert or a medication guide. The difference here is there's less exploratory data, and the data in the USPI and patient labeling focuses on generally the approved dose and dosage form from the phase 3 confirmatory trials. Next, let's talk about the ability to prescribe. Under an EUA, as we talked about earlier, it's not an approved product, so it can only be used according to the authorized use, which is discussed in the fact sheet. And this often comes with many limitations, and I'll show you that a little bit later. When a product has a full approval, a prescriber can use medical judgment to determine when to use a product in a given patient. And finally, the duration of product availability. Under an emergency use authorization, once that emergency is no longer declared, the emergency use authorization is then terminated and the product is no longer available unless we've achieved full approval. Once we've achieved full approval of a product, that's available until a sponsor withdraws the product or if some untoward safety event is identified. Next slide, please. So now that we've done a little bit of compare and contrast between an EUA and a full approval, I'd like to bring a little bit of this to life with the clinical programs that Lilly has run for our two major programs against COVID. The first slide shows the clinical program for our rheumatoid arthritis product, which we have repurposed to study in COVID-19. You can see on the right of the slide, there are 3 clinical trials that have been run. 2 of these have been run in conjunction with the National Institutes of Allergy and Infectious Diseases. So this is one unique aspect about our emergency use authorization journey. Typically, a sponsor will run their own clinical trials, especially until a product is approved for the first time. And in this instance, we've seen an unprecedented amount of partnership with the government to ensure we are running the right studies on an expedited timeline. So Act 2 and Act 4 are the adaptive COVID treatment trials. These are 2 arms in an overarching adaptive clinical trial that's being sponsored by NIAID, which is a component of the National Institutes of Health. And these both include baricitinib against different control arms. The third study is the Cove Barrier Study, or KHAA, and this is a Lilly-sponsored study, also with baricitinib, looking very similar to the ACT trials, but on a different background therapy. I draw your attention to the number of participants in these trials to highlight there are over 3,500 patients participating in these trials. So again, the data package that is being generated is not that different from what we see on a full approval. Next slide, please. This next slide shows the clinical program with our monoclonal antibodies. Again, we've seen unprecedented partnership with government agencies in the development of these antibodies. When we typically develop a drug, we start with independent clinical studies through each phase, and we move from phase 1 to phase 2 in patients and phase 3 to do our confirmatory trials. In this instance, it's been quite unique in that we've been able to compress those phases and actually run phase 2 and phase 3 in a single trial. So prior to starting Blaze 1, we did conduct a traditional phase 1 study, but then we embarked on the Blaze trials. And Blaze 1 has adapted from a phase 2 to a phase 3 trial. This has enabled us to do things very quickly because once we have the studies up and operationalized, we don't lose any time shutting a study down and restarting it. In addition to Blaze 1, we have Blaze 2, which is a study being done in nursing home patients. This study is quite unique from an operational perspective because typically clinical trial participants have to go to a site to be seen. And in this instance, we actually brought the site to them utilizing a mobile clinical trial unit, which was a retrofitted RV. It was a very creative approach from our clinical trials capabilities colleagues. While not depicted on this slide, we also have a study called Blaze 4. And Blaze 4 is being done in a lower risk patient population. And it's designed very similarly to Blaze 1 in that it allows us to add cohorts to the study so that we can continue uninterrupted but continue to test additional experiments with additional antibodies and lower doses. And for example, this morning we just announced an opportunity to test a third antibody in Blaze4 in partnership with Vir and GlaxoSmithKline. Blaze1 as well as Active 2 and 3 include our lead monoclonal antibody, bamlanivimab. And in Blaze1, we've also introduced etesevimab, our second antibody. The Active 2 trial is sponsored by NIAID, and it is very similar to Active 1 in the high-risk ambulatory patient population. Active 3 is in the hospitalized patient populations, and given early data from that study, we are no longer pursuing the research of our antibodies in that particular trial. Next slide, please. So based on the clinical plan that you saw and early data from Blaze1, we were able to receive an emergency use authorization for bamlanivimab in November of this year. Next slide, please. So bamlanivimab, as we've talked earlier, has not been approved, but has been authorized for emergency use to treat mild to moderate COVID-19 in adult and pediatric patients 12 years of age and older who weigh at least 40 kilograms with positive results of SARS-CoV-2 viral testing and who are at high risk of progressing to severe COVID-19 and or hospitalization. I read that authorization statement to you because it is quite extensive and is different than what we would typically see in an approved indication that doesn't provide that number of limitations. In addition to the authorized use, I'd like to share some important safety information about bamlanivimab. And the next slide, please. So as we talked earlier, not only do we share what is available about the product and the appropriate use, we share the limitations of the authorized use. And bamlanivimab is not authorized for these more severe patient populations based on data that has been generated from clinical trials. It's important that we make sure that we're transparent because we've not met the standard approval, the approval standard that we talked about earlier. Next slide, please. In addition, the important safety information is shared whenever we talk about the product. And as we discussed earlier, the limitations of the available data. So I won't read all of this to you, but I think it's important to call that not only are we putting forward the known safety information, but the limitations of that in this slide and my last. So next slide, please. And that's all I have for you today. Thank you.
Thank you, Dr. Ramseyer, for that wonderful presentation from Lilly. Our next guest, please welcome Dr. Suresh Mittal. He is a distinguished professor of virology at Purdue University. Please welcome Dr. Mittal.
Good morning, everyone, and thanks for having me on this important topic. For me, the disclaimer is that I have no financial interest with any of the product we are going to describe here and any of the company which I'm going to mention in this my presentation. Next slide, please. For emergency use authorization of the vaccine specifically, although the time period or duration for approval is pretty short, but compared to other product in this one, it still is going to go through stringent, you can say, safety and efficacy thing, because the phase 1 trial is important, phase 2 trial is important, phase 3 trial is important. And in that, we are looking for safety, that vaccine are safe to use and they are efficacious. So in this, for COVID-19 vaccine, the efficacy was that how many individuals got COVID in control group compared to how many individuals got COVID in the vaccinated group, and it comes out about 95% efficacy is there. And then we have to look for what are the potential risk and the benefit outweigh the risk, and then the product will get approved. So in this one, stringency is there. The only thing is missing is long-term efficacy, and the other thing is could be long-term side effect. And the safety monitoring system in US is pretty stringent, and there are various groups which work on that. But for this COVID vaccine, there was an expanded safety monitoring system, and that where the side effect of the vaccine can be studied at real time. So CDC has VSafe program where individual can report any side effect. And then National Healthcare Safety Network is also working on finding out if anything wrong with the vaccine or some individual get immunized. And in next slide, please. So difference between full approval and EUA approval. So as there for treatment and diagnostic, it is only there for public health emergency. Since it is a pandemic, so this is a right way to approve the vaccine through EUA rather than the regular system. And the potential benefit for these vaccines, they outweigh the risk, and that's why these vaccines are already available for human use. But only thing is limited is long-term protection and adverse effect, as I mentioned earlier. So full approval for any of these products, it's usually takes long time. And as for the diagnostic and for treatment, and even vaccine will take even longer time because the vaccines are used in healthy individuals, and because of that there is going to be a period which is going to be longer. Next slide, please. So when we talk about the vaccine, so there are conventional vaccine consider they are either inactivated virus vaccine or attenuated virus vaccine. But for COVID-19 vaccine, the vaccine which are going to be in the market or coming in the market, the first vaccine which came in the market was messenger RNA-based vaccine. So messenger RNA, what is that code for the spike protein, which is the most immunogenic protein, will provide protection. So once the messenger RNA encoded spike protein is injected in the system, it is going to the system is going to produce the protein and then develop immune response. The other kind of vaccines which are going to be available, they are vectored vaccines, especially adenovirus vector, and where the spike protein is being cloned in adenovirus, and then once the injection of adenovirus occurs, and that adenovirus is going to have the spike protein produced and then going to be immune response. And third one are protein-based vaccines. So the spike protein is being produced and then mixed with a specific adjuvant and then injected in the system, and then the antibody response is going to take place. Next slide, please. So the structure of the spike protein is very complex and its size is also very long. So in that protein, there are two cleavage site at S1, S2, and there is another one at S2 prime. And here is the receptor binding site, which is important because if the antibodies are there against receptor binding site, they are going to cause the virus neutralization. Next slide, please. So since two messenger RNA vaccine has already there for human use, so how these messenger RNA vaccines are manufactured and what they contain. So in short, the sequence for the spike protein will be here in the open reading frame. And before that, there are upper untranslated sequences which provide the stability to the RNA. Again, there is another untranslated sequence is a 3' also, that is also important for the stability. And then there is a poly A tail that is also important for, you can say, stability of the messenger RNA and its transcription. In addition to this RNA, a cap is attached. So there are different caps available, and based on that, that cap is also, you can say, it prevent the innate immune sensing. It also prevent the rapid degradation of the messenger RNA. Next slide, please. So first, we can see the differences between a DNA vaccine and messenger RNA vaccine. As some of us has more understanding about the DNA vaccine. So on this side, these are the DNA vaccine containing a gene of interest, and then there is a, you can say, lipid bilayer lipid membrane through which this can enter into the cells through endocytosis process. So once they are in endosomes, and after that, the gene will get released and then go to the nucleus, and from the nucleus, the transcription will occur, and after the transcription, there will be the messenger RNA will go to the ribosome, and it is going to make the protein. But in RNA vaccine, what is there? So once they get endocytized, so after messenger RNA coming out from the endosome, and it can go directly to ribosome and synthesize protein. And after the trafficking of the protein through secondary modification, then that protein can come on the surface of the cells and other things will happen. So in this one, it is bypassing the transcription process because the messenger RNA is already there. That is the major difference. Next slide, please. So how these vaccines work? In short, this part I have already talked about, the endocytosis and endosomal escape of the messenger RNA is there. So here I have also has the ribosome. So ribosome is going to make the protein. And once the protein is being made, that protein can be trafficked out of the cells or can be presented on the surface of the cells. At the same time, the protein, which is a new protein for the cells, it will also go through proteasome. And the proteasome will process this protein, and this smaller protein will be going to this endoplasmic reticulum, and through MSC class 1 antigen, they are going to present on the surface of the cells so that the CD4, CD8 cells can recognize this peptide and have immune response, which is of the CD8 T cell type or CD4 T cell type. So that other protein which is being on the surface or excreted out, that protein can endocytize in the same cell or different cells, and that will also undergo processing, and that protein will be presented on the cells with the MSC class 2 antigen, and this you can say recognition is going to lead to manufacturing of the antibodies. And that way we'll have a T cell response and the B cell response leading to the protection. Next slide, please. So when you look in the literature or on the online, then you will find there's a lot of myths and misinformation about messenger RNA vaccine or specifically COVID vaccine. So messenger RNA vaccine, one of the bigger myth is that vaccine can change my DNA, and which is not true, or any of the thing in here are not true. Other thing, people are saying that since these vaccines are being rushed, so they cannot be trusted. And it seems like none of these vaccines are rushed. Messenger RNA technology is around almost for 30 years. And the vaccine can lead infertility or miscarriage. That also not true. Vaccines are developed using fetal tissues, and that information is also not right. The vaccine can cause COVID-19, and some people are believing that side effects are more severe than if the person get COVID vaccine, or vaccine content are unknown. There's no benefit to individual who had COVID. And since vaccines are here, that the pandemic will disappear. So until people are going to take vaccine, the disease is not going to disappear. The vaccine may contain microchip or tracking devices to monitor people. That's also a false myth some of the individual are having. And then the vaccine will result in a positive test. That is also not true. It is only going to induce antibodies, and majority of the tests are based on PCR. So it is not going to do that either. Next slide, please. So if we consider difference between these two messenger RNA vaccine, which is already in the market, the Pfizer BioNTech vaccine, and the second one is Moderna. So age group are very similar. So it's 16 year, and the Moderna is 18 year above. Amount of messenger RNA is very different in this one. Pfizer vaccine has a lower amount, 30 microgram per dose, and Moderna has 100 microgram per dose. The difference between two doses in first one is 3 weeks, another one is 4 weeks. Vaccine efficacy is very similar. Side effect, they are very similar, pain at the site of inoculation. And a majority of us, we don't get any side effect. I got my first vaccine, and I got the Pfizer vaccine, and there was only a little bit of the pain at the site, and just for 1 day, and I did not have any other side effect. My daughter she got both the vaccine, means 2 doses of the vaccine, and she did not get any side effect. So majority of us may not get side effect, but some individual might get a little bit more of the side effect, but it's not a big deal. The storage conditions are different. Pfizer vaccine is stored at a much lower temperature, minus 70 degrees Celsius, and the Moderna 20 degrees Celsius, and both of them has modified messenger RNA, and their modifications are different. And both of these vaccines also contains a lot of lipid, and that's some of the lipid are different, these two vaccines. Next slide, please. And then there's a lot of salt there. And the other thing, in addition to the sucrose, is there in Pfizer vaccine, and there are acetic acid and the acid stabilizer are also there in a Moderna vaccine. Next slide, please. You might have heard that there are some cases has occurred where allergic reaction, and including the anaphylaxis, can occur in some individual. And this has happened in a majority of those individual has a history of allergy, because any chemical can cause allergy in person. So for that, what is happening, that when the vaccination are done, the history is being recorded. If somebody has a high degree of allergy, so they have to consult their physicians to get the vaccine or not. And it usually reaction occur within 15 to 30 minutes. And because of that, what is happening, that the individual has to stay in the facilities for that period of time. And if some individual develop, you can say, anaphylactic or high allergy reaction, they can be treated very well with epinephrine. So far anywhere else, we haven't heard that anybody has died because of getting the vaccine. Next slide, please. So the other group of the vaccines, they are adenoviral vector-based vaccine. In this one, the same thing is being done. The spike protein is being cloned into the adenovirus vector. So there are 3 different types of adenovirus vector has been used. Human adenovirus vector, which is type 5, and human adenovirus type 26. And the other one is chimp adenovirus based vaccine. So the Russian vaccine has first inoculation with human adenovirus 26 vector, and the second is human adenovirus 5. And then I'm going to talk about the Jensen vaccine contains only 26, and the AstraZeneca vaccine has only the chimp adenovirus. So after vaccination, what happens because of adenovirus, there is activation of the innate immunity, and where the dendritic cell and the macrophage get activated. And what will happen then, there will be development of the spike-specific humoral and cell-mediated immunity, including CD8 positive cells, CD4 positive cells, and there are going to the memory T cells and the memory B cells, so that the protective immunity is being introduced. Next slide, please. So the Jensen vaccine, which is the Johnson & Johnson vaccine as well, so has adenovirus 26 vector-based vaccine, and it seems like the results are expected anytime now, and so this vaccine more likely is going to get approved for emergency use sometime in February, and it is a single-dose vaccine, so that more people can get immunized. AstraZeneca or Oxford vaccine containing chimp adenovirus. In US, it seems like this vaccine will get approved in April. And this vaccine is already being used in other part of the world. In UK, people are getting immunized with this vaccine every day, and they haven't heard anything bad about it. In India, this vaccine is being used extensively. Brazil is using this vaccine currently extensively. So I think, in my opinion, this vaccine will get released also in the US. The vaccine, the protein-based vaccine, which is from Novavax, it is going to get released in the US, and it is under a phase 3 trial. They have little bit of the issue in some of the individuals who were enrolled in the vaccine. They dropped out because when this Moderna and Pfizer messenger RNA vaccine were available, so they were not sure they are in a placebo group or the vaccine group, so they thought it will be better to use this vaccine. And I think majority of those individuals who are dropped out, they were elderly, because elderly was a priority for the vaccine right now. Next slide, please. So some of the questions we can ask current vaccines, that does these vaccines are good for all segment of the population, because they have not been tested for children. They have not been tested for pregnant women. So those studies are currently going on, and I think there will be a separate approval to use in that segment of population. Another issue can be there is the protection is only from the disease that we know already, but does they also prevent from virus transmission? Those studies are also currently going on, and we will have a good answer pretty soon. And what is the durability of the protective immunity? And those studies are also going on. So I think within 3 to 6 months, we will have more information about all these 3 questions. Thank you very much.
Thank you, Dr. Mittal. That was very helpful and lots of great information. Our final speaker, and also our moderator of the panel, will be Ross Silverman. Ross is a professor at the IU Fairbanks School of Public Health, and he's also an expert contributor on law and ethics to the Indiana CTSI's COVID-19 reviews of relevant information and literature. Ross, please take it away, and then we'll have a panel discussion with all of the speakers and answer some questions from the audience. Thank you, Ross.
Thank you very much. And just as a first step, I want to take a moment to thank all of our speakers that we've had this morning, and just give a brief summary of what each of them, some of the insights that each of them gave us. Dr. Christensen talking about the multi-layered process, for review and approval through the EUA processes for treatments, therapeutics, for diagnostic tests, as well as for vaccines, talking about the fact that EUAs are not permanent. First of all, that they do expire when the emergency has come to an end. And also should point out the fact that emergency use authorizations can be withdrawn if there is new information that raises concerns about the safety and efficacy of those products. Dr. Parnas talked about EUAs and diagnostics, shared with us the various regulatory pathways that the FDA uses depending upon the risks associated with the particular devices, and the circumstances, the 4 circumstances that need to be fulfilled in order to go through the EUA process, as well as talking a little bit about the amazing compression of time that that they went through in coming up with their diagnostic assay development from 6 to 8 year process to a 6 to 8 week process. Dr. Ramseyer talking about the parallels and the differences in the requirements and the review processes for treatments under a EUA and for the full approvals. Talking about the limits that are in place on prescribing under an emergency use authorization, and also talking about the collaboration that they have had with the NIH in bringing together, bringing their monoclonal antibody emergency use authorization process and approval in place. Dr. Mittal talking about the strong and expanded safety and efficacy standards that have been put in place throughout the COVID-19 vaccine development and approval processes, talking about the continued monitoring of their safety and efficacy that is taking place. Also talking about where the limits are for what we are able to know at this point with the approval of the various vaccines, but debunking a whole bunch of myths that are out there as well. And also giving us a sense of what the differences are between the DNA and mRNA vaccines are, as well as what's still in the pipeline and should be coming forward in the next few months. I'm a professor, as you can see, at the IU School of Fairbanks School of Public Health. I am a lawyer by training, but my statement is I do not have any conflicts to disclose, and I am a lawyer, but I'm not yours. And so anything that I do say today during my presentation should not be relied upon as legal advice. If we can move to the next slide. I want to just talk for a couple of moments on the policy arena and sort of the what's next components of the rolling out of the vaccine under an emergency use authorization and what that means for society. We really, I think, can safely say that there has been no scientific breakthrough that's been more eagerly anticipated in recent history than the development of a COVID-19 vaccine. It is a novel pathogen for which to create a vaccine, and the 10-month time period that it took from when we first identified the pathogen to when the vaccine was authorized under the EUA has blown away all previous records for the speed of developing a safe and effective vaccine. The prior record was for the mumps vaccine, which had taken a 4-year process to go through all of those various stages. What we've heard from the other speakers as well is, the emergency use authorization can be used in a wide array of different circumstances, and at the same time, this whole emergency use authorization process is only a 15-year-old process. And so there is, some ongoing, and really there hasn't been this much pressure on a review process as there was, or as there has been during the last 10 months. And we can say that from a perspective of, because of the enormity of the issue being faced by society, and also because of political pressures as well. The emergency use authorization was first used for a vaccine. It was actually used in order to approve the anthrax vaccine so that it could continue to be in use by the Department of Defense as a preventive measure versus possible biowarfare agents. We know through this emergency use authorization process, we do have strong safety and efficacy review standards throughout the vaccine development process. The median follow-up for the phase 3 data that had to go into this review process had to be at least 2 months long. One of the things we do know with regard to most vaccines is that a vaccine adverse event is most likely to show up in the first 6 weeks after its administration. So we know that we do have a strong sense of the kinds of the majority of side effects that might show up, but there are still, of course, unknown unknowns given the fact that we are now presenting this vaccine and delivering it into millions of people, and we do now have a longer time frame at which to look for potential effects. We know that we now do have the 2 vaccines that have been approved, Pfizer for those 16 and older, Moderna for populations 18 and older. You might ask, why is one for 16 and older and why is one for 18 and older? Again, as far as the emergency use authorization process is concerned, that is directed in part by the request from the manufacturer. Pfizer asked for an approval for population 16 and older. Moderna asked for an approval for 18 and older. And so that's really the difference. It was the fact that Pfizer asked for some minors to be included in the vaccine approval process. There is, again, high safety and efficacy here. And these are now being delivered into society. With regard to the emergency use authorization, resulting in broad shares of the population getting vaccinated, one of the next questions that frequently has arisen in my discussions has been, will we see mandates associated with these vaccines? Well, first and foremost, the emergency use authorizations require that fact sheets be shared. And in those fact sheets, it states explicitly a portion that is included in the law, which says that people do have a right to refuse these vaccines. However, that doesn't necessarily mean that people have a right to be free of consequences for refusing. There may be circumstances where employers end up making decisions that for the safety of the other employees and the safety of the working conditions, as well as the safety of the people with whom their employees interact, that there may be some circumstances where vaccines really would become required. I would see this potentially most likely in healthcare settings. And we can talk during the Q&A if people are interested, or if you want to follow up with me at another later point as far as the ins and outs of potential mandates. I think there are better ways for us to be going about encouraging and getting vaccine uptake than that, making it as easy as possible for people to access and say yes to, and by engaging in the community with populations in order to answer questions as much as sharing information. Next slide. We talk about these vaccines as being a threshold moment in our COVID response. It's, however, not a silver bullet. As we know, this is not going to be a cure-all for the epidemic. We do need to rely upon a huge array of different safety measures that need to be not only continued but reinforced as vaccination gets out into society. Vaccines are one part of a systems response. And this Swiss cheese model is a nice way of looking at and breaking down all the different places that we need to put precautions and safety measures in place, some of which are personal, but many of which are systemic and shared responsibilities in order to ensure the safety of the population and to bring the spread of this virus under control. This is arguably the responding to this outbreak is probably one of the most complicated problems that we have tackled as a society. A colleague was recently quoted stating that to study COVID-19 is not only to study the disease itself as a biological entity. What looks like a single problem is actually all things all at once. What we're really studying is literally everything in society at every scale, from supply chains to individual relationships. And the problems that and the challenges that we are facing in trying to bring this under control, we can see run across everything from the RNA, all the way up to massive societal issues like racism and injustice. Next slide. We do know, again, that vaccines themselves, if they're not getting into people's arms, they don't stop anything. So it's the vaccination process that actually is going to be stopping diseases. And what we also know is that there is hesitancy out there. We need to ensure that we are engaging. We are not just presenting this as a solution, but we are engaging populations, answering questions, as Dr. Mittal talked about earlier, debunking myths, but also recognizing that people are concerned about their health and safety and concerned about the health and safety of their loved ones. So we have to, we have to tread carefully. We have to engage with communities rather than in just merely informing communities. And we have to break down as many barriers as possible to make it as easy as possible for people to get access to the vaccine. Next slide. Again, how we go about doing this, we are now starting to see steps taken to get the vaccine out and distribute it to the general population. Every state has been given free rein as to how they are going to construct their process. Every process is going to have its challenges. I mean, I think one of the challenges that we face is we are creating entire new systems of delivery in a 24-hour news cycle and social media system. So it's almost in many ways, I think, like finding out about a shark bite someplace in the world. If there is an issue that arises, if a decision is made and somebody disagrees with it, if priorities are set and because there are only finite numbers of vaccines, somebody is one group is prioritized over another. The group that got deprioritized or was not selected is going to raise questions and concerns. No one is going to be 100% happy 100% of the time as this process goes forward. And so you have to make sure, as we're working through these challenges, that you are being guided by core ethical principles as far as ensuring access to care, as far as equity is concerned, as far as minimizing barriers, as far as giving people information, and as far as setting the priorities for vaccination, while also recognizing that we are rolling this out while also trying to treat everybody in the hospitals, while working with oftentimes under-resourced health systems and under-resourced public health systems. And so it's an unbelievable logistical challenge to be watching rollout in real time. Next slide. We do see that in the last couple of days, a national strategy for response and pandemic preparedness has come out, and that included in that, the second goal in that process is a quick, effective, and equitable rollout of vaccination. Currently, I think one of the benefits and one of the challenges we've had with regard to emergency use authorizations and the COVID-19 vaccines is that the Pfizer and the Moderna vaccines, needed to show that they were safe and efficacious, and they cleared that bar by miles as far as their demonstration of safety and efficacy. So I think we on the one hand have really strong products that we can present to the public, and at the same time, because of the storage needs of those first products, it did also create some new challenges during that rollout of both manufacturing, supply chain, cold chain, and the sites at which those first could be rolled out to. I think we are scaling up and will be moving exponentially faster with vaccine distribution as we move into the next few months. Next slide. But the biggest thing I think that is going to be as a next step in order to ensure that the vaccines are taken up, we do have to address issues of vaccine hesitancy. I think ultimately, as more and more of the population does get vaccinated, more and more of the population will be willing to get vaccinated. That said, I think we also have to recognize that perhaps some people didn't want to be the first. And so they're waiting to see what happens as the first wave of folks get vaccinated. We do need to make sure that outreach for vaccination information and the processes through which people can sign up is as localized as possible, using as many different mechanisms as possible for people to allow them to sign up. There are a whole lot of people out there who don't have good internet access, who don't know how to use the internet. So having an entirely internet-based sign-up process is going to leave them behind. So we have to make sure that we can reach those folks in other ways. Indiana has the 211 system that people can call into, as well as their website for people to sign up. And still, I think that's going to, present its own challenges. We need to make sure it's available in as localized a place as possible. We need community leaders from within the various communities speaking in language that the local communities understand to be on board as part of this process as well. Trust in public health, there's an old saying, arrives on foot and leaves on horseback. And we need to make sure that we are able to, and we were already dealing with significant trust issues as the pandemic response rolled out, with regard to histories between public health and certain populations. And so we need to be careful and be open and communicative with these populations, with varying populations, in order to make sure that those who are most vulnerable are also going to be equally protected in this process. I'm happy now at this point to bring in, I'd like to invite the other panelists to unmute and to re-reveal themselves so that we can engage in the Q&A with the other speakers. and with our audience members. Again, you can put your questions to the panel into the chat. And if you don't have questions, I have a few that I can use in the meantime. Okay, so the first question here, recognizing that there may be long-term side effects still remain unknown, what is your advice to those who may be vaccine hesitant due to concerns about potential long-term side effects?
So I can take this one. Everybody can hear me?
Mhm.
Okay. So the reason is I don't expect that the vaccine will have long-term side effect, because the vaccine component will be gone from the system pretty quickly. So once they are not there, whatever we can see within a month after the second inoculation. And if we are not seeing any bad effect during that period of time, we don't expect that we'll see later on. So I don't have any concern that long-term side effect can be there.
Thank
Donna,
you.
we get that question quite a lot from people who come to our vaccine clinic, and we try to emphasize that we have many months now that since these vaccines started to be given to people. So as time goes on, we accumulate more and more data, and we haven't seen any long-term problems with any of the patients.
Another audience question. What would each of you like vaccine providers to tell their patients that might help increase uptake?
Well, I could speak as being the clinician in the group. I think the most concern that people have about these vaccines is that they have this perception that they were rushed into use. So one of the things that we try to emphasize to them is the fact that all the knowledge that we have about coronavirus and the immunology that is necessary to protect people has evolved over the past 17 years since SARS-1. So as we start discussing SARS-1, and then we start talking about the coronavirus, the MERS virus that popped up a few years ago in Saudi Arabia, I think people start getting a little bit more relaxed as, oh, well, people have been studying this for a long time, so it doesn't appear to be as rushed. So they tend to relax a lot more. And then we talk about, we have to be upfront that, yes, potential side effects do occur acutely, but there's no long-term effects. And I think that usually, if you explain those two things, I think people tend to, they tend to be more receptive to the vaccine. The other important thing that we talk about is, especially when we're dealing with African-American communities and Hispanic communities, is that we need to emphasize that this virus causes a lot more morbidity and mortality in their groups. And it's key that they get vaccinated as a way of protecting their family and friends and neighbors. And I think they tend to, if you can go and have a conversation rather than just looking at a brochure or a piece of paper, I think that people tend to have their questions addressed, and I think they tend to be more receptive.
Sorry, I can add another thing. I think if the healthcare people, they share their own story and their family story that they got the vaccine and how did they feel about it, I think the patient could be more receptive receiving the vaccine.
Absolutely.
And then one more thing that I wanted to add perhaps is that really getting COVID-19 really has really serious long-term effects that we still don't know yet. There's a lot of knowledge that it's coming up. The term long COVID on these patients that have long-term consequences. So that's another point of view to highlight the benefits of the vaccination to actually prevent from having going through the disease and potentially having to end up in the hospital. So,
and towards this end, one other piece that I would just bring up is that one of the pieces of information we don't really know well yet, and it reinforces the fact that even when people are fully vaccinated, they do need to still socially distance. They do still need to mask. We know that this is very successful in helping people avoid hospitalization and avoiding death due to COVID-19. We still have little information about whether this prevents the vaccine from being passed from one person to another. And so that's one of the factors I think people need to keep in mind, and why we need to make sure we still have in place strong adherence to all the other preventive measures that we talked about, in addition to vaccination, which is, again, seen as a really strong step to stopping death associated with this virus.
So I'm going to jump in here because we are at the hour. We have one additional question from Marie that I'd love to have our panelists respond to. But before we do that, in case some folks have to sign off, I just want to thank our speakers very much for all of these insights. I learned a lot. Remember that this will be available on our website. And I want to go ahead and let's go ahead and answer that question. And then we want to also tee up that our next frameworks will be happening on February 18th. But let's go to Maria's question, if we would, Ross, if you'll facilitate that.
The question related to vaccines for children, what you expect the timeline for vaccination approval in children, and do you expect that to be going through the regular process or an emergency authorization as well?
Well, I think no question that that is data that we need to have, and many of the vaccine companies are now enrolling children down to 12, and then after we have that data, they'll start enrolling even younger than that. The important thing is to know about the safety and how effective it is. In the very young, it's very important to know exactly what the precise dose is going to be. So I think that has to be studied as well. So it's going to take several more months before we have more data in regard to the 12 to 16 years of age. However, the good news is that since we have a lot of data that is accumulated with adults, perhaps for that age group, we don't have to enroll 30,000 participants to be able to show that the vaccine is efficacious as well as safe. So I would say we're probably looking at at least my guesstimate is I don't think we're going to have very much to offer to young children until the summer. And it will probably go as a EUA because it's the quickest way to get the vaccine into the arms of children.
Yeah, I agree.
Emergency use authorizations cannot be mandated by the state. So if people have questions about whether that's going to lead to a child vaccination mandate, that won't be coming under an emergency use authorization.
I agree.
All right. Well, I'm hearing that. JoBeth, I think, Patty, we're up.
I want to thank everyone on our panel for sharing their insights today. And I also want to thank our audience for joining, and hopefully we've been able to share some information with each of you that you'll be able to share with others. And again, I want to highlight that we have our next frameworks will be on February 18th. We're shifting topics for that one. And we'll have our esteemed colleagues from Purdue, Notre Dame, and Indiana University to share how they do tech transfer in our efforts to make sure that Indiana-based innovation gets out into the world. So please, the registration link is up on the chat right now. Thank you so much to everyone for joining us. Sorry that we're a few minutes over, but it was certainly informative. So we'll see you next time. Thanks.
Thank you very much.
Bye-bye.
Thanks,
Thank you.